Hi, everybody. Welcome to GenBio Live. This is our regular broadcast discussing recently published work in the Journal of Microbiology and Biology Education. My name is Rachel Horak. I am staff at the ASM Education Department, and my work involves programs for educators and researchers in professional development. Today's GenBio Live, we're very excited to bring you a paper called Community College Biology Students Understanding of Vision and Change Core Concepts. Our speakers today will feature Noah Courtney at Texas Tech University, Michelle Smith and David Esparza of Cornell University. Our Q&A, our question and answer period today will be moderated by Sarah Fankhauser. She is the GenBio Research Senior Editor. The Journal of Microbiology and Biology Education is for science educators all over the world, and it is open access. You can read all of our articles free of charge. We're currently soliciting articles for a new special series called The Practical and Positive Uses of AI in STEM Education. Check out GenBio's website or scan the QR code to submit and learn more about this exciting special series. Next month in San Antonio, the ASM is going to assemble the Conference for Undergraduate Educators. It is an annual conference now in its 32nd year. And we will be gathering from November 21st to 23rd in San Antonio. And it's not too late to get yourself registered and join us for 2.5 days of learning and connecting with educators from all over the world. So with that, I'm going to turn it over to our GenBio Senior Research Editor, Sarah Fankhauser, for an overview about the section. Sarah.
Thank you so much, Rachel. I am thrilled to be here today and have the authors of the paper, Community College Biology Students Understanding of Vision and Change Core Concepts. So this article is in the research section of GenBio. The goal of this section is to share new knowledge about a particular area or subject, teaching approach, or student learning and perceptions. These papers are hypothesis-driven investigations, which include rigorous assessments of teaching, delivery methods, educational approaches, activities, student learning. And I'm so excited to have the authors here to talk about their paper. I'm particularly intrigued by this article because I teach at a 2-year institution at Oxford College of Emory University, and I appreciate the attention to students in these 2 years of their college experience. So I will hand it over to our authors so they can tell us about their project and their process of research and their findings.
Thank you so much. So I'm going to go ahead and share my screen. If I can get the presenter view to work. Great. Okay. Let me see here. So hello and welcome. Thank you all for attending today. And thank you especially to the folks at ASM and GenBio for hosting us. And thank you, Dr. Frankhauser, for moderating. My name is Noah Courtney. I'm a first-year PhD student at Texas Tech University. And today I'm really excited to talk with you about research that I conducted as an undergraduate student exploring community college biology students' understanding of vision and change core concepts. And I want to start this off with an interactive activity and ask you all about your connections to community colleges. And to provide some background on community colleges, these are 2-year publicly funded centers of educational opportunities. And I'm really interested in how we all connect and the variety of ways in which we connect. So if you could write those in the chat, we'll take a quick look. Maybe you're a student or an instructor, or maybe you're an instructor at a 4-year institution and you've taught a community college transfer student. So I'll give that a second. Seeing teaching at a community college, I'm seeing instructors from 2-year colleges and also from four-year universities and teaching transfer students. So this is great. I I am also very connected with community college, and before I talk about our research and about the paper, I want to share how community college really has impacted my life and influenced this research. So I'm a proud community college graduate, but my story didn't start there. When I was 19 years old, I failed out of college and I enlisted in the US Coast Guard. And after a four-year military career, I really wanted to give college a second try. And I enrolled at Harford Community College in my hometown in Harford County, Maryland. And I know that there are some folks from Harford Community College here today, so here's a shout out to them. Um, Harford is where I took my first biology course when I wasn't even a biology major. But I took this course with Professor Madden, and she introduced me to the idea that I could be a scientist and introduced me to research as a second career option after the military. While attending community college, I looked for research experiences, and Harford supported me in going to the University of Alaska Southeast for one semester, where I trained to be a scientific diver, and I gained my first research experience in kelp forest ecology. And shortly after that, I came back. I graduated from Harford in December of 22. And after that, I transferred to continue studying biology at Cornell University. And this is where I learned about biology education research. I joined Dr. Michelle Smith's lab, where my mentor, Dr. Dave Esparza, encouraged me to really follow my own research interests. And as a junior year transfer student, I set this goal of wanting to write an honors thesis. And I wanted to make sure that that honors thesis really focused on community college biology students, students like me. So the work that I'm about to share is my first author publication, and also served as a successful undergraduate biology honors thesis at Cornell. So when I started this project, I jumped into the literature and found that community colleges are unique. They enroll a higher percentage of students who are first generation students who come from low socioeconomic backgrounds, who are student veterans like me, and students from underserved and minoritized racial and gender demographics. Community colleges also make college accessible. They offer lower tuition costs. There are often open admissions policy, and they provide a source of education that's close to home with built-in support systems for students, both socially and academically. I was also surprised to see just how many undergraduate STEM students attend community colleges. Over 40% of undergraduate STEM students in the US have attended community colleges, whether that's taking a single class or pursuing an entire associate's degree. But I was equally surprised to find that within the biology education research field, the literature primarily focuses on four-year students, and only about 6% of the literature primarily focuses on community college STEM or biology students. And it led me to the question, how can we best serve this population of students? And I was asking this as I was formulating an idea for an honors thesis. So as I got farther into the literature, I found the Vision and Change report. And this report was published in 2011 and serves as a guide and a framework that unifies what biology students learn in undergraduate degree programs over the course of their 4 years. And they established that students should learn these five core concepts: evolution, structure and function, information flow, systems, and pathways and transformation of energy over the course of their undergraduate degree. And in response to that report, the GenBio-MAPS assessment was created and published with the goal of measuring students' understanding of these 5 core concepts. And this assessment is open source. It's available online. Students take the assessment online, and they take 15 multiple true-false items that span across these 5 core concepts that you see on the screen. And those items also span across all 3 major areas or subtopics of biology. So when a student completes that assessment, a few different metrics are provided. One of those is the overall score, a student's complete score in the assessment. And there also are 5 scores, one for each of the 5 core concepts. And also the assessment ends with a short demographic survey. And previous implementations of this assessment, specifically one in 2019, was very large. They administered this to over 5,000 students across 20 institutions. And I'm going to come back to some of the more specific results of this prior study later. But it's important to note that students scored highest on items measuring systems understanding and scored lowest on items measuring students' understanding of information flow. But what really stuck out to me in this prior study is that 90% of their sample were students' responses from 4-year institutions and only 10% from 2-year institutions, which made me interested and ask the question, what can we learn by centering community college students? And how is community college conceptual understanding in biology really shaped? So we asked these research questions in our paper. We asked, how do community college biology students perform across the 5 vision and change core concepts? And we asked, do institutional, course, and student level variables contribute to variation in community college biology students' conceptual understanding? And to explore these questions, we analyzed results from 601 community college student responses to the GenBio-MAPS assessment. And these were collected over the course of 5 years and spanned across 9 institutions. And the demographic makeup of our sample aligns well with what we typically see in community college STEM contexts. So to address our first research question, we really wanted to find out across these 5 scores that each students have, are these scores similar? Are students scoring similarly across these 5 core concepts, or are some of them coming easier to students, or students finding some of them more challenging? And to explore this, we used linear modeling, but to really make sure we were asking the question in the best way, we wanted to make sure that we controlled for variation caused by nestedness. And nestedness causes variation because students learn in different ways in different contexts. And when students are at different institutions, they learn differently. And the same goes for when they have different instructors or are in different classes. So using statistical analysis, we found that the variation between institutions and between classes was great enough that we should control for that in our model. And after we did that, we have some results. And first, we found that across the evolution, structure and function, and energy and matter core concepts, there was no statistically significant difference between these 3. And on the y-axis, we have our percent correct on the assessment. And on the x-axis, the similarity in letters indicates that there's no difference between them that is significant. But we also found that students scored significantly higher on items that measured systems. So systems is defined by vision and change as the ways in which living things are interconnected and interact with one another. And students scored better on these items. We also found that students scored significantly lower on items measuring their understanding of information flow, the influence of genetics on the control of the growth and behavior of organisms. So once we had these results, we were interested in digging a little bit deeper, and we wanted to know how students were scoring on the biology subtopics that are also measured. And what we did is within each set of core concept scores, we looked at, are there differences between student scores on these subtopics? And interestingly, in information flow and systems, the two core concepts where students scored significantly higher on systems, significantly lower on information flow, we saw this same trend where students were scoring lowest on the cellular and molecular and highest on the ecology and evolution. But importantly, that these were not consistent. They were consistently inconsistent. And we see this kind of across the board. We found that students on multiple core concepts scored lowest on cell molecular subtopic items. And also that students scored highest pretty frequently on ecology and evolution subtopic items. When we look at the items that students scored the lowest on, we see this trend reflected. If you look at the core concept column of table 6 here, we see that 4 out of the 5 items measure students' understanding of information flow. And when we look at these knowledge statements, we see a couple consistent themes pop out. That these items relate to the central dogma and gene expression and protein synthesis, things that heavily align with that cell molecular subtopic. So now our second research question, we wanted to explore, do institutional, course, and student level variables contribute to variation in community college biology students' conceptual understanding? And to answer this question, we created a model that best explains the variance in overall score. This model also helps us identify any significance between minoritized and non-minoritized students within each demographic group. So, for example, if there was a difference in overall score between first-generation students and those students whose parents had attended college, this model would tell us that. So similar to our first research question, we wanted to account for nestedness and the variation that it causes. And through analysis, we found that there was enough variance between institutions, between instructors, and between classes that these were variables that we had to control for. And after that, we used a method called backward selection, and we identified the most optimal model to explain the variance in overall score, and that included multiple groups of students. So in which of these groups did we see a significant difference in overall score? And the answer is 3 of those groups. We found that men scored higher than students who did not identify as men. We found that students with non-minoritized racial identities scored higher than students with minoritized racial identities. And we found that students in life science majors scored higher than students who indicated their major was outside of the life sciences. I know that that was a lot of results and a lot of graphs, and I'm really excited for the conversation surrounding this question. What do these results mean? And to start, what I want to do is put our research findings in context with that previous study, which focused primarily on students at four-year institutions. So when we look at conceptual understanding trends, we see a lot of similarities. We see that in both contexts, four-year students and community colleges, we see that students scored higher on systems items and scored lower on information flow items. And we also see the same trend in those subtopics where students scored higher on items situated in ecology and evolution topics and lower on items situated in cell molecular topics. But where we see the difference between primarily the primarily four-year study and our community college study is at the student level differences. When we look at the student level differences in overall score, we see different trends between our study and the previous study. We see that in our study, there's less disparity than there is in the 2019 study in four-year contexts. And we also see that notably, there was no difference between first generation and continuing generation students in our study, and that there was no significant difference between students whose first language is and is not English. So to summarize our conclusions, community college biology students are building conceptual understanding similarly to introductory students at four-year institutions, and community colleges may be creating unique positive infrastructures for students who are English language learners and who are first generation. But the question is, what should stakeholders take away from this work? And I want to take a minute to recognize the fact that there are a lot of stakeholders when it comes to community college education. There are instructors, there are advisors, there are all of the people who make community college STEM programs run. And this also includes researchers in our field. But I want to start with educators. So first, I think this emphasizes the fact that educators can evaluate core concept coverage at the course level using the GenBio-MAPS assessment. And as I had mentioned before, this assessment is free and available online. I'm going to put all the QR codes that are going to come up in the next few slides at the end of the talk. But this QR code leads to the publication of the assessment, in which there is a link. Also, educators can utilize open access educational resources that are aligned with vision and change. And I think that these 2 points really go together, because if you were an instructor and you were to find that your students are maybe struggling in information flow, and specifically within the cell molecular subtopics, it's possible for you to utilize these open access educational resources and go in and find things like lesson plans and other tools that can support your students' learning in these specific areas. And a recent publication by our colleague, Dr. Kira Trebergs, highlights the fact that over the past 20 years, the resources available have expanded massively. And in this recently published manuscript, she provides links to multiple different resources that are available. And lastly, if we take it a step broader and we look at the design of majors or the design of curriculums, the Vision and Change report is still openly available, and curriculum alignment with Vision and Change can further support students' growth and development in conceptual understanding. So when we think about what researchers should take away from this work, there are a few key points. And first, implementing community-based participatory research is really important. And community-based participatory research is a framework where instead of researchers who are doing human subjects research are looking at their participants in a separate as a separate entity, they're focused on building relationships and really making sure that everyone is contributing to create the best research possible. And I think that building relationships between 2- and 4-year institutions is the best solution to support community college biology student learning. Additionally, our study looked at conceptual understanding specifically at the introductory level, but there are still open questions about how this conceptual understanding is changing over the course of a 2-year degree program, and also how that conceptual understanding changes through the transfer process. And there have been multiple recent calls about the need for more research investigating student transfer, and I think this ties in nicely with those recent calls. Lastly, our research explored a very small subset of the multitude of factors that influence conceptual understanding. And here I note that non-academic factors and psychosocial factors could also be explored in their relationship with conceptual understanding. But in community college biology students specifically, these non-academic factors might look like having a family or having a job and the intricacies that come along with that. And I think that these are areas of research that are open and ready for research to be done. I think these are really important. And I think that this work opens up a lot of doors for potential future directions. So with that, I want to thank first and foremost my 2 mentors on this project. As I had mentioned before, this was work that I completed as an undergrad, and I was learning how to do research as I moved through this process. So I can't thank Doctors Dave Esparza and Michelle Smith enough. There are QR codes to the different resources that I had mentioned here. And if you don't get the chance to get them through these QR codes, feel free to send me an email and I'm happy to share them. A big thanks to our community college participants, to all of the people who supported this research, and personally to the Harvard Community College mentors who inspired this research from the get-go, from my first day as a scientist. So with that, I think I will pass it back to Dr. Frankhauser for the Q&A.
Great. Thank you so much, Noah, for walking us through the research that you all did and your findings. This is fantastic. So just a reminder, please put your questions in the Q&A. That way I can see them and we can facilitate asking and answering the questions. So I will start us off. At the end of your paper, you provide some tips for actually implementing the assessment, or not to, but you recommend implementing the assessment in the introductory biosequence, which I loved. And, for me as a researcher and as an educator, I often read educational papers, and this one just really stood out as one that I could implement almost immediately, and I loved it. Do you have any tips in terms of for educators who want to implement this? Like I'm thinking like practical, like timing or like um I guess like how to communicate it to students, I guess the value of taking this and what we could do with the data afterwards.
Yeah, that's an excellent question. And I think an important mark would be that this assessment is taken online and at home most of the time. So while that can be a benefit to educators, it also comes at a cost of something around 45 minutes to students at home. So having that discussion with students and saying, here's the easy access online. However, this is going to take you time. And also, I think that showing data like those that I have just shown could be really beneficial, and showing students, I'm not just giving you this assessment because I'm going to take the results and whisk them away and never and not share the impact. I think sharing the impact with students uh, is a big benefit of using this assessment.
Yeah, I love that. I didn't even think about sharing the outcomes with students. I'm writing that one down. Okay. We definitely have questions in terms of how the assessment is operationalized, and in the paper and I think you mentioned this in your talk, that it's a bank of true or false questions. And I believe it's in table 6 that you have some sample questions, particularly with the cellular molecular subtopic, which is the field I teach in. So I was very excited about this. And I just want to read one question because it did make me think. And I was like, damn, if I had to take this, now I understand why it takes 45 minutes. Okay, so it's different cell types have the same genetic composition. With some exceptions, cell type specific gene expression patterns are not caused by programmed loss of genes during development. So I can see just how nuanced these true or false questions can be, and how they are intended to make students think through every bit of the question. But when I'm looking at these questions, I'm curious if you know how the original assessment tool was validated. And I ask because I'm like, if I were to take this, I think that I would have to struggle to really think about what the question is asking. So I can see where it would be challenging for students as well.
I think that we are really fortunate throughout the entirety of doing this project that Dr. Michelle Smith was a major driver in this, and I'm actually gonna popcorn that question over to her if that would be okay, because
Sure. Thanks.
she has good insight and played a key role in the assessment development.
Yeah, thanks. So it's a really good question because for any of these assessments, there's kind of 3 things we think about. Does it align to the big standards like the vision and change standards? So the questions align there. Are they valued by instructors, and do students understand them? So we spent a lot of time aligning back to vision and change to make sure we had coverage throughout. For the instructor part, what we would do is we would interview instructors at the beginning with questions just like the one you read and said, what do you think the answer is? And then once we had a set that we had iterated a little bit, we would send out surveys to instructors at all kinds of institutions, so community colleges, four-year institutions. We would ask them to answer the question, and then we would ask them to give us feedback on the questions. And as you can all imagine, instructors have a lot of feedback about the questions. So we would gather it from numbers, and we would look for patterns, and we would make revisions there. And then the last key is, did the students understand what we're asking? So we spent a lot of time in what we call think aloud interviews, where one of the researchers sat with one of the students, and the students would read the question aloud and answer their thinking. And we're looking for this magical moment where in just a second we can tell if the student has the right answer for the right reasoning or the wrong answer for a common misunderstanding. So even the first part that you talked about, the genetic material being the same in cells, that's a common misunderstanding we know from other practices and other research studies. And so we were looking to see how they would explain it. And I often describe it as the snappy moment where they're just starting into their explanation and we know if their explanation's matching their answer. Hope that helps.
Yeah, that helps so much. Thank you.
And one last thing that I would like to add for the GenBio-MAPS assessment, I think what's really amazing about it is that since it follows this multiple true-false structure, it allows for us to kind of to uncover partial thinking, right? So like students have some correct ideas sometimes, and students might have less correct ideas of other things. And so by giving students this general statement, like you were reading, Sarah, and then having them think through these sub-statements that are asking them if it's true or false, you can see, okay, well, maybe a student has a correct idea here, but then they hit this next one, and it's like, oh, maybe a little less correct. And so, yeah, it's a really good way to capture the complexity of student thinking.
Thank you both. So I know that this question has actually come up a lot in conversation about this paper. So I'm sure that you all have really dug into this and thought about it and have very detailed answers. But is it so thinking about the timing of the survey at a community college versus a four-year institution, and granted, it is meant to be at the, I guess you said at the beginning of the introductory sequence and at the end of the introductory sequence, and that you're taking a snapshot, which is great. Is there any way to compare that to the four-year population, or yeah, I mean, I guess that's really the question. Is that assessment or comparison possible, or is that a future direction, or maybe not. Maybe it's a different direction.
I think that's a really good question. And as far as what we saw, the scores that we saw on the assessment are comparable, very similar to what we see in the introductory results from the original GenBio-MAPS assessment implementation. And I do think that while the GenBio-MAPS is designed to be at the beginning and end of an intro sequence and at the end of a degree program, I do think that there could be utility in implementing at the beginning, middle, and end of a two-year degree program. But at the same time, I think that implementing it in that way at community colleges also has drawbacks and difficulties. I think about those students who come in and they take one class and then they might leave for a year. Part-time students aren't necessarily on that same degree track where you would think, oh, well, the students in this course are all at the same point in their degree program. And that's just simply not the case. So I think there are open directions for that. But it's a really good question. I'm not sure if Dave wanted to.
I think the only thing I would say, thank you, Noah. Yeah, I think the only thing I would say is that I would love to have a study that has it, that implements it at the beginning time point or right before a student takes their introductory course sequence, both in 2 years and 4 years, right? And then at the end of that 2-year period, both at 2 years and 4 years. Again, our results are this mixture of 600-ish students from 11 institutions, kind of all across the range of taking their first bio course at their community college, right? And so for some students, it was right at the beginning when they started this course. For other students, it was right at the end that they finished their first course. And so it really provides a snapshot of what students are learning. But I think there are ways to standardize that collection. I'm hoping one day that our study adds some detail into what students know at community colleges and how we can improve bioed in the context. But I also think that, yeah, there's some future work to be done in standardizing that time point aspect.
Great. Thank you. So a question came in that is somewhat connected to one of the questions that I posed earlier around tips for implementing this assessment. But do you know if the students were compensated at all in terms of taking this assessment, or would you recommend that? Like, and I would certainly not pay my students, but I might give them extra credit.
As far as I think the answer is it depends. I think that's up to the instructor who's implementing. Every student individually consents at the beginning, and of course, we remove participants who are not yet 18 years old or who don't consent to their assessment being used in research. But I do think that, I mean, of course, in all human subjects research, it's important to make sure that students are appropriately compensated for their time, whether that is an extra credit or payment. But I think it depends on the institution and the instructor.
And then
Great.
something, oh, Michelle.
Go ahead, go ahead. Oh.
Yeah, I was just going to say it's most often participation points. So their students won't be scored for being right or wrong. They'll be scored for participating and trying the survey out. And I know sometimes it can be hard to keep track as an instructor if you've got hundreds of students in front of you about who's taken or not taken. So one little trick I tried when I was teaching my genetics course is I would say things like, if 75% of you do this assignment, everybody will get the points. And then I can just keep track of the numbers and then get really close to that number and give everybody a few extra credit points. So that's one way to help, because I know everybody's really busy keeping track of lots of students these days.
Absolutely. And the last thing I would share is, so in 2021, there was actually a study looking at the test-taking motivation of students who took the GenBio-MAPS. And just like Michelle said, the main recommendation there was offering participation points at when you implement the assessment, because it's long, right? And it's cognitively, it can be cognitively demanding, right? They're really strong but challenging questions. Noah and I took the assessment several times while we were doing this work, and it's an amazing tool, but it can be challenging, right? And so offering students that incentive can help combat those effects of test-taking motivation, and also provide educators with a better metric of what their students are learning in their courses. And I linked that study in the chat as well.
Thank you. And David, you just made me feel a lot better about me kind of having to sit and think with this one question for a minute.
Noah and I would sit there. We would be in my office. At the time I was in a small grad student office, and we would just sit there in the office and be like, oh my God, like this is hard. So yeah, offering those points really does make a difference, because it's a really well-designed tool.
Yeah. Well, thank you. We have a few more questions that have come in. So I'm just actually going to start from the top. So how many students were part of your population? And so in the question, did you actually reach out to the students, or were you able to get the data from the larger study?
So these data were I want to say passively collected, since the open publication of the assessment in 2019. So we could say that our population size was in the range of 56 to 57,000 responses. And that's not just from community colleges, that's from all responses that were ever collected from the assessment. And what we did is we went through with the most recent data available and ran our filtering protocols to pull out the community college responses that, of course, met appropriate criteria. And that is how we ended on the number of 601. And we also we got rid of multiple responses. So if a student had taken the assessment multiple times, say once in their intro class, and then a different instructor administered it in a future class, we only looked at the first time the student took that assessment.
Perfect. Thank you. And David oh wait, sorry, one more follow-up to that. So do you have any sense of the response rate? I guess, is it possible to determine that?
I think it's hard to determine for certain, because it is open source, and because if this was intentionally applied, say, at a single institution and was implemented at a biology or a STEM department, then it would be fairly easy to track how many instructors gave this to how many students, and then how many responses did we get back that would be a fairly contained study. But in the case of our study, we were looking at the full set of responses that came from the open access implementation and publication.
Awesome. Thank you. So the next question, David, you answered in the Q&A, so thank you. But I want to ask it out loud because it could be of interest to others. So do you think this tool would be useful for a smaller sample size, for example, class of 20, which I myself teach on that order. So I would love to, to hear more about that.
Dave, it seems like you had some thoughts on this, so I can let you take that one.
Absolutely. Yeah, it totally is a tool that you can use in smaller classes, and folks regularly implement it in small classes. So I would say for a few of even the courses that we had collected data, or that data had come from, so to speak, tended to be around I would say this is a rough estimate, but like 20 to 50 students, right? And what's really cool is you can get metrics on students' understanding of each of these core concepts. If you request a link through the link that I posted in the chat, what that process really looks like is completing a course information survey. So saying, I teach this course. I need the survey for this date. Here's kind of why I'm interested in implementing it. And then you get sent a link automatically to the survey. You implement it. And then eventually, after the survey closes, you're sent a report that includes the names of the students that took it and also a summary of your class's performance. And if I remember correctly, you can also disaggregate based on demography. And other characteristics like that. And so it's a really cool way to it's a cool way to understand your students' learning, especially again, kind of regardless of what class size you're teaching.
Great. So our next question is, do you have any insight on why in community colleges there's less disparity in terms of first generation students? And I think this is in comparison to the study by Couch et al.
Yeah, that is an excellent question. And I want to preface that by saying, off the top of my head, I don't have any literature to cite, but I have hypotheses, and I have my experience as a community college student. While I'm not a first generation student, I have many friends who are, and the smaller class sizes, the higher I would say the accessibility of professors and mentors and advisors in the community college system make it easy for first generation students who may not have grown up with all of the information about college to go and have multiple touch points and multiple resources at a community college that can support them in being a college student, especially during that first year. Compared to a university, I think universities tend to be large environments, and sure, there are a lot of resources, but I think at a community college, in my experience, it's been easier to find and access resources and people who can point me in the right direction. My transition from community college to Cornell was a big jump. And in my experience, I found that it was easier to find resources at my community college than it was at university. There were just a few extra steps required. So that's my hypothesis.
Thank you. For those of us who may want to implement this assessment in our own classes, do we need IRB approval?
I'm happy to take that one if that's okay.
Yeah.
So the assessment and the collection of the data, you don't need IRB approval. So there's an IRB form out of Cornell, and students can say if they want to consent or not consent to having their data included in research. I will say that if you are using something like GenBio-MAPS to make improvements in your own curriculum or in your own classrooms, you can kind of think of it as something you're not planning to publish with the external world, that in fact, no, you don't need an IRB because you're using the information for improvement. So that's just one thing to keep in mind. But it's always good to have a conversation if you're unsure with your local IRB office early before you start something, and they can often answer their questions. And if they have questions, we can also put them in touch with the Cornell IRB office too.
Fantastic. Thank you. Noah, I'm going to ask you to speculate again. So, as someone who teaches in the molecular and genetics realm, I'm curious if you can speculate as to why these were consistently scored or had the lower scores, not just in your study, but also in the Couch study, and why systems seem to score the highest across both.
Just realized I was muted. I can speculate, but I can try and do one better and share some really awesome insight that I gained from K through 12 science teachers at the National Association of Biology Teachers conference back in 2023. Yes, that's correct. I presented an early version of this work at that conference. And that was a question that I had. Why are we seeing these trends where students are scoring lower on cell molecular items? And the K through 12 teachers said, this makes a lot of sense. And that's because ecology and evolution topics are taught and discussed from such an early age in K through 12, where you're talking about plants and animals and nature. And students have a very tangible touch point with those topics from very early on. And as you get more into middle and high school and you start learning about cells, these things are abstract. You can't go and hold a cell or a mitochondria in the same way that you can maybe a snake that one of your teachers has. And there's been some really cool work done on visual representations of DNA, of chromosomes, and some of those findings show that there are a lot of different ways to visually represent these things, but we don't always teach students all of those visual representations. So I think it's really easy to misconstrue some of those things when we can't physically hold them. Does that answer your question?
Yes, it does. And you were able to answer my question, even though I phrased it incorrectly and said something about systems when I meant, and you understood that I meant ecology and evolution. So thank you. All right. See if there are any remaining questions. Well, we have just a few minutes left, so I would love to end this with you all talking about how you work together, especially mentoring an undergraduate through educational research and then through the publication pipeline, which I think is a pretty unique experience for an undergraduate. So I would love to hear about that process.
To what I can provide to answer this question, I want to emphasize the fact that when I joined the lab, I asked Dave, how do I be a biology education researcher? And the first 2 to 3 months, he said, you're going to read a lot of literature and you're going to familiarize yourself with the people and the work in the field, along with building quantitative skills. I had never used RStudio or coded before. So by starting with learning to read literature really consistently and really thoroughly while building my quantitative skills, I think that set me up early in the process for success. But beyond that, I will let my mentors take the wheel from there.
Yeah, I mean, so I would say my approach to mentoring generally is just that that it's a bi-directional relationship, right? So like as I'm not a strong believer in hierarchy, and I think because of that, when I work with Noah, he's my colleague, right? And his ideas have just as much pull and weight as any of my other colleagues. And so I gave him a lot of flexibility. I mean, you had a lot of flexibility in this process, right? And like Noah got to decide, okay, well, I'm going to put this predictor in and see how this works. I'm going to try out these models. And a lot of it was just providing that kind of agency, but also I think for Noah, to some degree, right? Like we have this massive database of responses that are generated through the GenBio-MAPS, right? And so when Noah came in, I was like, okay, well, what do you want to study, right? And Noah talked a little bit about this in his first few slides, right? But Noah's motivation was really, how do I give back to this community that contributed to my success? And I think I mean, there's broader frameworks on like community cultural wealth and thinking about how to bring students' background into the work they're doing in research, right? And so that's a lot of how I frame my work is what does a student think what is important to the student themselves, and how can I help them bring that into the research process? So it gives them some ownership, it makes it fun, it's something that they care about. And then again, also like I think Noah and I just had fun as we worked on this. I mean, he would call me or text me and just be like, "Hey Dave, here's how this is going," or or this didn't work, right? And just really, I mean, I tried to structure that process and make it fun and make it and also just try and demystify it too. Publication can be such a uh, process, right? And so I tried as hard as I could to just say, okay, here's what, like, here you should email the editor and say, hey, is this a good fit, right? And you should, like, here's how you make tables, and this is generally the norms of the field. Also, just kind of saying these things that I think tend not to be written down, or discussed, and just really explicitly saying, this is kind of what the field is like.
Yeah.
Dave brings up a really good point, in that you asked specifically about like getting through the publication process, which I think, from my experience, a lot of my undergraduate peers at the time were like, publication is a hurdle. That's a really hard thing to do. And I remember Dave posing it as, well, we can't think about publication until we write, right? We can't think about publishing a paper until we learn how to write the methods, until we learn how to write an introduction, and then make that introduction very specific to the work that we did. And I think learning how to research write is such an important skill to learn as an undergrad that I think the biggest part of this research process, the biggest point of growth for me was writing. I went from being an okay writer to a better writer, and I'm getting better every day. But if we can research right, then publication is meeting requirements of journals and making sure that our research is really rigorous. But I think it's a stepwise process that naturally will lead to publication if all those skills are built in the correct order and over time.
Well, thank you so much. I'm glad that GenB could support that process for you.
Thank you.
They absolutely did, and we appreciate it.
Great. Well, any final thoughts? Because I see Rachel's back on, so she's probably going to tell us to wrap things up.
I think the last thing I would say is just if you're curious about using the GenBio-MAPS, there's a few links in the chat, and also we're just really thankful for you, Dr. Finkhauser, and Rachel and Dr. Horak for hosting us and letting us talk about this and this work, and also yeah, thank you so much.
Thank you.
Right back at you, David. And thank you so much, author team, for joining us. I'll make a quick note to say that I recognize a few people from Hartford Community College in the audience, and they would be so, I bet they're tickled pink and so proud of you, Noah. So thank you very much, authors, for joining us. A quick note to say that our next GenB Live webinar will be Friday, December 5th at 1 p.m. Eastern Time. And the topic, this is really exciting for those of us education researchers, will be a recently published article called Tools for Coordinating Measurement Use in Science Education and Training. So the website will be updated within a week or so. You can go register yourself, and like all GenB Lives, make them free for everybody to attend. So thanks so much for joining. You can watch the recording on our YouTube playlist, And we hope to see you at another GenB Live soon.
Thank you for having us.
Thank you. Thank you so
David.
much.
Thank you.